actually. One you can call it as the karyokinesis part, and then followed by the cytokinesis part. Under karyokinesis of mitosis, yeah, that is the right way to say that. Under karyokinesis of mitosis, what we saw is it can be further divided into four phases. That is prophase, metaphase, anaphase, and telophase, right? So the prophase was the first phase, right? And it is the longest phase. So you have to remember this, the longest phase in metaphase is the prophase. So what did we understand by prophase? We understood that there are certain things that is happening in the prophase. The first thing is it happens after the completion of G2 phase, right? And then the chromosome material, basically it's like thread. All of it, which is like randomly in a thread-like structure in the nucleus, they have to be condensed, and the condensation of chromatin material take place, or the chromosome materials take place. So all of them get untangled, and they start getting rearranged very properly, right? And then all these chromatids, they come and attached at the centromere region, right? They form this X-shaped body. And now what happens? These chromosomes undergo, what do you tell? Hold on, reorganization. And more importantly, I told you that in the beginning of the prophase, the nuclear envelope starts disappearing. Along with nuclear envelope, you can expect some of the cell organelles. So I told in the last class that some of the cell organelles will disappear. Yeah, your friends are here. So can you guys tell me what cell organelles disappear during the prophase time? Yeah. So these cell organelles disappear, and I want you guys to answer. So if you remember that, it's well and good. It's a very important topic. I hope, let's see if you guys remember that. Okay. So, and then we mark the end of the prophase, seeing that there is no nuclear envelope or the nuclear membrane anymore. And then we have this centrioles arranged at the opposite poles, and they have started forming the spindle astra or the spindle fibers, right? So the spindle astra or the spindle fiber formation, okay? See, the formation of spindle fiber itself is at, hold on. If this is your, right, centrosomes, or centromere, sorry, what do? They produce these spindle fibers like this. So they have this nice, what do you tell, like fountain-like structures. So like complete thick fiber-like structures at opposite poles. So this thing is called as spindle astra, okay, or spindle astra. So after the formation of this, we can tell that we are going for the next phase, that is the metaphase. Now it is clearly distinguishable. Metaphase can be very, very clearly distinguishable compared to that of your other phases. By what? You know that by these, the nuclear, the chromosomes have been very well condensed, and these guys are ready for the next phase. So they align at the center, or it is also called as a meta center. So they come and align at a meta center, and they align in a plate, or we also call it as metaphase plate. So they form this plate called as metaphase plate. By now, your spindle astra would have completely developed, and now they try to attach to the chromosomes, right, at the kinetochore region. And we talked about, some of you guys asked, what is the difference between kinetochore and the centromere, right? So we talked about it. So once they have attached to the kinetochores, this is where we had to talk. So now anaphase. See, the role, the idea is to separate the chromosomes. Now that the kinetochores have been hooked by the spindle fibers, now the anaphase starts. So what happens during the anaphase is the centromere splits. See, you can see here, the chromosome is an X-shaped structure, and it is held by the centromere, and they are made up of chromatids. Okay, so these chromatids, when they're pulled each other, so they get separated. Okay, they move towards the opposite poles. And by the end of anaphase, what you can see is these see, this is a very well clearly distinguished phase. I can tell, if you see in the microscope, you can distinguish anaphase very, very well, because you can see that the chromatids have almost moved towards the opposite poles. All right. So by the end of the anaphase, you can say that they have completely migrated towards the poles. So that should be the end of the anaphase. By the end of anaphase, what should happen? You would have the spindle astra, and spindle astra would have done its job, or the spindle fibers would have done its job. Now the disappearance, I mean, like, once they have come to the opposite poles, there is no use of the spindle fibers anymore. So once they have reached that point, okay, the spindle fibers detach from the kinetochores and they start dissolving. They don't disappear, they disappear. Let's say that they disappear. So they start disappearing slowly. So you can see that the disappearance of the spindle after forming by the end of the anaphase. And you can also say that the chromosomes have reached the opposite poles. Now coming to the telophase, the last phase of your mitosis, karyokinesis. So what is happening here? The chromosomes have reached the opposite poles. They have done their job. I mean, like they have reached their destination. Once they have reached their destination, what you can see is, guys, the cell in the beginning, let's say that it is like this. Okay. Now, by the time it has come to the phase, which phase, the telophase, you can see that the cell is already preparing for something called a cytokinesis. I mean, while the telophase is going on, you can clearly see that the cell simultaneously is preparing for something called a cytokinesis. Where is your nuclear material now? All your chromosomes have reached the opposite poles like this. Once they have reached the opposite poles, yes, the packing is done, moving is done. Now, what should be done? Unpacking should happen. Okay. Now, what did I tell? By the time you see the chromosomes have reached the opposite poles, guys, you can also observe that the cell is preparing for cytokinesis. That's what I say. Okay, well, let's keep that on the other corner. You just have this idea, keep this idea in the back of your head that when I look at the anaphase, my cell is looking something like this. There is a small furrow I can observe, okay, in the telophase. Now, these chromatin or chromosomes are which are highly condensed when they have reached the opposite poles. Now the unpacking happens. What is unpacking? The decondensation or disassembly of these chromatin fibers will take place. So once this happens, all your very well organized chromosome bodies, they start becoming the thread-like unorganized structures again. As this process is taking place, now more importantly, what has disappeared? The nuclear membrane had disappeared. So this nuclear membrane which had disappeared will start reappearing again. Okay, will start reappearing again. Now, I'm surprised that you guys did not ask how does it disappear and how does it reappear? Okay, who does this job? The job is done by Golgi apparatus, guys. Okay, if you want this information, the job is done by Golgi apparatus. Golgi apparatus actually synthesizes cell membranes. Okay, remember this particular fact. So now the reappearance of nuclear membrane is taking place. And along with that, I asked you a question, but unfortunately, all you guys are sleeping. Welcome back to the class again. So I asked you a question: which of the cell organelles disappear in the beginning of the prophase? So I told you that Golgi apparatus, endoplasmic reticulum, mitochondria, and nucleolus. Nucleolus, okay, is also part of nucleus. All of them, very, very important, guys. Nucleolus, Golgi complex or Golgi apparatus, and endoplasmic reticulum, including mitochondria, they would have disappeared during the beginning of prophase. Agree? Now, by the time we have come to the telophase, they start reappearing again because the cell is already dividing. Got it? Now, since the spindle fibers were already disappearing, the spindle fibers would have completely disappeared. Okay, they have gone, and the centromeres also will disappear. By the end of the telophase, what will happen, guys? Centromere will disappear along with the spindle fibers. Got it? Now, this marks the end of your telophase. By the end of the telophase, how does it look like? I can see my cell that the furrow is already developing very fastly like this. I will tell you what is a furrow, but you just keep that in your head. The word called, the groove or something like, when you press two things, it's just coming towards each other. So this furrow is coming towards each other. And now nuclear membrane is quite well developed. All your chromatin material is quite highly decondensed. It is highly decondensed. The Golgi apparatus and all your cell organelles like endoplasmic reticulum and mitochondria would be very well be formed. Okay, they're very well formed. And now that marks the beginning of the cytokinesis. Cytokinesis, I told you, it is a cytoplasmic division. Correct? No. See, the cytokinesis, even though we are talking about general cells, we have two major different cells. What is that? The cells with cell walls and the cells without cell walls. Do you agree with me, guys? Yes, yes or no? Yeah. So can I tell that the cells with cell wall and the cells without cell wall, their division, especially their cytoplasmic division or the cytokinesis, is different from each other? Is it different or not? Yeah. Yes, no, guys. Yes, yes, absolutely. It's quite different. Now, what is happening here? So it's a little bit deviation. Okay. In your textbook, the general cytokinesis is given. We will go on to differentiate what would happen if there is a cell wall. How would the cytokinesis take place? This might be your in your head. I will cue that in. Okay, I will just come to that part. See, guys, now generally how the cytokinesis happens. The cytokinesis happens by the formation of a furrow. What is this furrow? It is called as a cytoplasmic furrow, which is formed with the help of a plasma membrane. Okay, see what is happening, guys. Your cell is like this, and there is a plasma membrane across it. So now the cytoplasm along, you see, the plasma membrane starts moving towards inward. So this is called as invagination. Invagination means what? Just going sucked inward. So as the plasma membrane gets sucked inward, the furrow starts developing within the cytoplasm. Okay, the cytoplasm at this stage, what you can see is it is not as dense as you see in the normal time. In the normal time, when the cell is highly functional, no, the cytoplasm is like a jelly. It's very dense. You can see the jelly can be very loose and very dense. So it's watery and less dense during the cytokinesis when it is happening here. Got it? No? So when the cytoplasm is very dense, the furrow can move further and further and further. So the plasma membrane, as it forms the furrow within the cytoplasm, now the furrow touches with each other. The plasma membranes will cut at a point, and then the cells will separate. That marks the end of your cytokinesis, forming two daughter cells. Got it? Now, this is a very general idea of how the cytokinesis will take place, and it happens exactly the same in all animal cells. I can tell the animal cells don't have cell walls, correct? So in us, it will happen like that. So let's go and look at the plant cells. Our plant buddies, they have a very, very rigid outer structure, and this outer structure is called as what? It is called as a cell wall. And a cell wall is so hard and it is a dead structure. It is dead. Cell wall is dead. So it cannot tell the cell wall, see, come alive, form a furrow. It's not possible. So for that, what happens? Your Golgi apparatus is there, no? So the Golgi apparatus will start bubbles. Let's say that this is your cell, and this is where the division has to take place, guys. Okay, let's assume that this is where the division has to take place. Okay, here it is. So let's assume that here the division has to take place. Now imagine that this cell has a cell wall. Okay. So now to divide that, what will happen? Your Golgi apparatus forms bubbles, and these bubbles we call it as vesicles. Uh, Vesicles. If I'm wrong, the spelling, please correct me. E or I don't know. Vesicles. So Golgi vesicles we call it as. So these vesicles are just put like bombs in a line, in a proper line. They synthesize these vesicles. You get my point? What is the point of these vesicles? These vesicles form something called as hemicellulose. Okay, which is flexible cell wall. Your cell wall is made up of cellulose. It forms a hemicellulose wall. So these vesicles, they start reorganizing, means they start becoming functional, and from the center towards the outside, they start forming the wall. Okay, they start forming the wall. Until when the wall formation will take place? Until they touch the plasma membrane and beyond. Now, this will form the very primary cell wall. Okay, very, very basic cell wall. After they have touched these ends, this will start becoming thicker and thicker. So once they start becoming thicker and thicker, now these cells will separate out. It will just, the cytoplasm will be separated out like this. And this hemicellulose, I told, no, hemicellulose. So this is hemicellulose, like a sandwich. And it will be replaced by a layer of hard cellulose on either side, like a cementing. When the hard cellulose starts forming here, now the two plant cells, very thin layer of cellulose starts depositing, and now the two plant cells will separate. That will mark the end of cytokinesis in plant cells. So who helps the plant cells in their cytokinesis means you should tell it is Golgi apparatus. What do Golgi apparatus will do? They produce Golgi vesicles. What do Golgi vesicles will do? Golgi vesicles will start forming a hemicellulose furrow. So this furrow is formed by who? Golgi apparatus. Guys, did you understand? Yes or no? Can you confirm, please? Yes. If you guys can't chat, you can just give a thumbs up. Okay, that will also do. Okay, thank you. I think Rakshita is one that, yeah, she responds all the time. And where are the other guys? Please come back because the next topic is quite very important and I want your attention. Okay, so chalo. So the next quite complicated topic is the meiosis, and some of you might feel it easy, some of you might feel it a very long topic to understand, but it's very what do you tell, systematic. So I'll try to make it as easy as possible. So I hope you have the complete attention. Okay. So what is meiosis? Does meiosis happen in all organisms? So that is our question. So let us say that we have an organism, and the organism has a cell, okay? And let's say that its genetic material is having only n number of chromosomes. When a genetic material is having only n number of chromosomes, we know that what do we call it as? We call it as haploid. Haploid, correct? No? So we have organisms, we call it as haploid organisms, correct? No? Okay. Or we also call it as haplontic stages. Okay, we also call it as what? Haplontic. So my question for you guys is, what is haplontic? Or we say the generation of alteration of generation in plant kingdom. What is haplontic life cycle? What is diplontic life cycle? What is, yeah, one if I get, I'll understand that you guys know the concept. Let's see. Haplontic means N. That's correct. That's correct. Okay, guys, I will ask you one more question. Let us take that I have algae. Okay, I have algae. Okay, hold on. I have algae. You will understand this so clearly now. 2N. Diplontic is 2N. That's correct. Okay, all right. So algae. Tell me, if I see an algae, what kind of a body it has? Whether it has an haplontic body, a diplontic body, what it is, guys? What kind of a body it has? Major body is what? If I check the algae cell, what should I find in their cells? N number of chromosomes or 2N? N. Absolutely correct. So they are haplontic. They are haplontic. Major plant body. Like me, if I check my cell, what do you see? You see that all my cells are made up of diploid number of chromosomes. Where can I find my haploid cells? Only when I produce the germ cells. Okay, only when the germ cells are produced like an organism like me, who's a diploid guy, I can see the cells haploid only when I produce the germ cells. My haplontic life cycle is limited to the formation of sperm or ova as a human being, correct? No? But if you see the plants, in the plants also, the algae's main body is what? All the cells are made up of haploid cells. Now, when they have to go under gamete formations, even they have to reproduce. No, yes, they undergo asexual reproductions, but they also form gametes. Asexual gametes and sexual gametes also they form. We know that, right? So when they produce gametes, what kind of divisions they have to go? If the plant body is haploid, what kind of divisions they have to undergo for the formation of gametes? You guys tell me. Any okay, I will just check the YouTube guys also last time they were answering. Okay. Uh Anything? No. Hold on. I want your opinion because we have to interact to understand. All right. N says N. Um, okay, I will say. If I have already a haploid body, okay, the body is what? Haploid. And I have to produce what? Gametes. So if my body is haploid and I have to form gametes, what kind of divisions will I undergo? I will undergo mitosis. Why mitosis? My cell has only one set of chromosomes, which I call it as N. In our biology, there is only room for N or 2N or 3N also. But it cannot go below N. Why? Because N is the complete set of chromosomes. You understand my point? So below N, there is no way possible. Okay, it maintains the chromosome. Exactly. It has to maintain the number of chromosomes. That's why in all haploid organisms, during gametogenesis, they undergo mitosis. Why did I say this? Because when we discuss meiosis, no, when we discuss the significance of meiosis, we tell that meiosis is a type of cell division that happens during the formation of gametes or formation of germ cells, etc. So I am telling that it is not always the case. It is the case. Meiosis is so significant when you have to reduced, yeah, exactly. When you have to sexually reproducing organisms, when you see sexually reproducing organisms, most of them are diploid. Okay, most of them are what? Diploid. So when they are diploid, they have to undergo their number of chromosomes is what? 2N. So they have to produce sexually. So that's why when they undergo meiosis, they produce haploid cells. These haploid cells are formed by meiosis. Okay, so that is a simple fact. Now, I cleared the small doubts, which usually, yeah, which people usually get confused. So always meiosis means they are only responsible for the formation of gametes. No, it's not the case. It's not the case at all. You can just see the different organ. If you just read the plant kingdom, and if you understand something called as alteration of generation concept, I think you guys studied this. It's called as haplodiplontic, haplohaplontic, diplodiplontic. So these life cycles come in your plant kingdom. If you read that, you will understand. Oh, okay. It depends on the plant, or it depends on the organism. Okay, we didn't study that plant kingdom. All right, we will take one class on alteration of generation and we'll understand. It's important, but let's see. Okay, chalo. So now let's come to meiosis. The general name for meiosis is called as reductional division. The reason why it is called as reductional division is because if the parent is deciding to go under meiosis, by the end of meiosis, it produces four cells. And four cells will have the chromosomes number as n, that is from 2n to n. That is called as half the number of chromosomes are distributed to the daughter cells. Okay. Now, this is one thing. Now, generally, very generally, in all higher organisms, meiosis occurs during gamete formation or gametogenesis. Okay. So, like you can say spermatogenesis or oogenesis, or you can also see in your plants formation of pollen grains, formation of egg cell. For that, what do you need? You need meiosis. Okay. Now, what happens here? The key feature is that meiosis is quite complex compared to that of your mitosis. Okay. Meiosis can be divided into two stages. One is called as meiosis 1, and the other stage is called as meiosis 2. Why it is called as meiosis 1 and meiosis 2? Because the cell division occurs twice here. See, one cell is going under division to form four daughter cells. No, it is not a one-step process. So it is going under two-step process. So how does it do? One cell first divides into two, and the further two cells again divides into two more cells. So this is a complete division. It has to go through this and through this. Only then the meiosis is complete. Okay. So that is why this first stage is called as meiosis 1 and the second stage is called as meiosis 2. So I said, take a small diversion here. Can you guys see that the beautiful star-like structures here at the poles? Okay. This is called as the spindle asters. So when you look at in the electron microscopy, they look magnificent. Okay. So like an aster formations. So that I just wanted to tell you this. So let's say, so meiosis happens and the result is the formation of four cells. Correct, right? It is a formation of what? Four cells. So if it is forming four cells, so in a cell cycle, we understood that the cell undergoes G1 and then S, G2, and then we have M phase. Now, during the S phase, the DNA replication happens. That's what I told you in the last class. After the DNA replication happens, the DNA is doubled. Once the DNA is doubled, we observed in mitosis that, okay, the DNA is doubled. Now the condensation of DNA happens. We have the chromosomes. Yes, it is, since it is doubled, we can just separate them. Correct, right? So this is your idea. Now, since it is meiosis and the division is happening twice, that is meiosis 1 and meiosis 2, does it mean that the DNA replication has to happen twice? No, guys. Only one time the DNA replication would have happened. That is especially during the S phase. So after that, there is no DNA replication. Next, what will happen? Only organizing and division. Only organizing and division. Let's see how it happens. So what is the things that happens in meiosis? What are the significant things that is happening in meiosis? The most important thing that happens in meiosis is, first thing is, I will tell you. I asked you, the last class, I talked about what are sister chromatids and what are chromosomes and what are homologous chromosomes. Now my question is, what are homologous chromosomes? What is the meaning of homologous chromosomes? What is homologous chromosomes, guys? I'm sorry if I'm eating the time, but I can finish, but small basics. No? What is homologous pair? Let's see. I will just jump in because the time constraint. Okay. See, guys, I have one. For example, we all have 23 pairs of chromosomes, correct, in each cell. So among 23, the total number of chromosomes would be 46, 23 pairs or 46. Let's say it is 46 for our easy understanding. Okay, 1, 1, 2, 2, 3, 3, 4, 4, 5, 5. I call it a pair because I have a left shoe on the right shoe. So I can't wear two different shoes. So if I have a left shoe, I need a right shoe. Okay, one. Okay, there is the first one from mother, second from father. That is called as a homologous chromosome. That idea of mother and father's distribution is not correct. See, guys, actually, Rakshita, that answer that you told, one from mother, other from father, is called as a homologous pair. Okay, there's a small mistake. First, what you told, one from mother, one from father, please tell that it is wrong. Okay, leave that. It is wrong. Okay, I'll tell you why it is wrong. Because it is me. I am one organism. Am I going under meiosis? Yes. So where is the mother? I'm single guy. I have to produce what? I have to go under gametogenesis. Any human being will undergo gametogenesis. So it is my chromosomes that is being divided. No, so there is no mother or father here. But in our cells, what is happening? The chromosome number is in pairs, like the left shoe and the right shoe. Okay. So how we can arrange like this: 1, 1, 2, 2, 3, 3, 4, 4, 5, 5. So this 1, 1 is called as what? Homologous pair of chromosome, or it is also called as homologous chromosomes. Got it? No. This 1, 1 is homologous chromosome. Sir, if I take 1 and if I take 2, what do they become? They become non-homologous pair, or it is also called not pair, it is called as non-homologous chromosomes. Got it? This is called as homologous, this is called as non-homologous. Now, what are sister chromatids? I told you our chromosome, complete chromosome, is made up of X-shaped structure. And in the X-shaped structure, there is a center, and this center is called as a centromere. Now, why the centromere is present? Because since it is an X, there are two thread-like structures involved. If I cross two thread-like structures, it becomes an X. So each thread-like structure is called as what? Sister chromatids. Got it? No? These things are very must for you to understand anything in meiosis. Is it clear? Right? Okay. And don't mind you about your wrong answers, whatever happens. So it's all right. Everybody has an understanding, just needs a correction. Clear, guys? Now what happens here? Let's see how the what did I tell you? First, the pairing of homologous. Very important thing is pairing of homologous chromosomes will happen. Pairing means what? Wherever they are distributed, they're all brought together. So 1, 1 be together, 2, 2 be together, 3, 3 be together. So this is called as organizing of chromosomes. So when they organize, they are arranged in pairs. So once they're pairing, like similar kinds of chromosomes are organized. Next, what happens? Recombination will happen. I'll explain you what is recombination. Okay, so the next important process in meiosis is what? Recombination. Pairing of homologous chromosomes and recombination are the two major steps in meiosis. Simple and plain. Okay, so let's see what happens. We are going to the process of meiosis. When the meiosis starts, okay, when does meiosis start? After the replication of chromosomes. Okay, the chromosome replication has happened. Okay, and now the chromosome has formed. Now, once the chromosome has formed, okay, by the end of S phase, now we go for the meiosis. Meiosis can be divided into same like mitosis. What is that mitosis? Mitosis can be divided into what? Prophase, metaphase, anaphase, telophase. Okay. Similarly, meiosis 1 can also be divided into 4 phases. What are those? Prophase 1, prophase 1, metaphase 1, anaphase 1, telophase 1. Got it? We are all putting it as denotion as 1 because since it's happening in meiosis 1. So the most complex part in the whole meiosis process is the prophase 1. The longest and the more complex process that ever happens in whole of meiosis is prophase 1. Very easy to understand. Only thing is you have to have the chronography. What is chronography? Like Avengers 1 and Avengers 2. Okay, you don't watch Avengers 2 and you go for Avengers 1. Similarly, you have to follow the order of events. To follow the order of events, you have to remember the names. So let's see, guys. The prophase 1, which is the first phase of the meiosis 1, it can be divided into several substages. Okay. What is our several substages? Why do we divide it into several substages? Because the most of the cellular events remain the same. Most of the cell, it looks the same, but the chromosome is quite differently behaving at each stage. So based on the chromosomal behaviors, we can divide this into substages. What are our substages that is happening in prophase 1? We have something called as liptotene, and the second stage is called as zygotine. Third stage is called as pachytene, and the fourth stage is called as diplotene and diakinesis. LZPDD. Okay, what should you remember the sequence? L, Z, P, D, D. What is L? Liptotene, zygotine, pachytene, diplotene, diakinesis. You can understand something called as kinesis. So put it in last. Always remember liptotin is the first stage. Always remember liptotin is a first stage. So you have to remember the names in sequence. Liptotin, zygotine, pacatine, diplotin, diakinesis. If you remember the names in order, then the series of events also become ordered. So let's see each of these. We'll try to remember things also with the names. Okay. First stage is liptotin. What's happening in our cell? The cell is going under prophase 1. Remember, it's going under what? Prophase 1. Here, it is since it is the very beginning of the prophase, the nuclear membrane, okay, your plasma, the nuclear membrane slowly has started its, disintegration process. Slowly, the nuclear membrane is starting to disappear. I'm not telling that it is full-fledged. It is starting. It is starting. That's all I can say. And as usual, your other cell organelles like Golgi apparatus, endoplasmic reticulum, as well as the ribosomes, they also have started to disappear. So this is the two basic things that we carry from the mitosis. Now, next, what happens, guys? Very important. You have DNA. That is your genetic material. Correct? No? You know that the beginning of the prophase is all about condensing of this genetic material. When the DNA is condensed, what did I tell? These long thread-like structures are reduced to short thread-like structures, and these short thread-like structures are called as chromatin. Here it is, chromatin fibers. Okay. So once they're forming into chromatin fibers, they are very, very long. I mean, like long, I can say long. Slender means very thin. Okay. They're becoming what? Long, slender, thin structures. Till when they become condensed, they become so condensed that the chromatin fibers are more condensed, more condensed. They're long now. They're long now. Why they're long? Because it is not so tightly packed. Okay. It is quite loosely packed eventually to become like X-like structures. What is our X-like structures called as chromosomes? Got it? No? In the liptotin stage, what is happening? We know that the prophase 1 is all about condensation of the genetic material, like in the mitosis. Here also condensation of genetic material will take place. So by the end of the condensation, you can see that the chromatin fibers have been formed, and these chromatin fibers are organized into long, slender chromosome-like structures. Okay, remember this? Long, slender chromosome-like structures. Got it? And now here what happens? You have the nucleus. This is your nucleus, and the nucleus is becoming more and more flaccid. Means it's not dense, it's becoming loose. So that is why the nucleus will also start enlarging. The nucleus is enlarging by the end of liptotin. Clear? Next, let's go for the next stage, that is zygote. What happened, guys? By the end of the liptotin, we have already achieved something called as X-like structures. Correct? No? Now, just imagine this is a cell and things are very randomly organized. And in human cell, let's say that like X-like structures are around 46 in number. And within the small space called as a nucleus, they're just here and there. By the end of what? By the end of liptotin, all these chromosomes are just scattered around. Now, what is happening? It has to be reorganized. How do we reorganize? All the see. Let's assume that like this. Okay, like this. All your chromosomes are lying here and there. Now it has to go for the next stage. What is our next stage? It is called as a zygotine stage. Now the chromosome behavior becomes quite, unique. What do they do? Less in so they condense even more. Once they become condensed even more, now they will start organizing with each other. They will start organizing. What are they organizing into? I told you already. The chromosomes are deployed in number, so they start pairing with each other. How can I pair? If I have a left side one shoe, I have to pair with the same type of shoe on the right leg. Same thing is happening. If I have one chromosome, it has to pair with its partner. What is its partner called as homologous pair? So it pairs with its 1, 1, 2, 2, 3, 3, 4, 4. All of these guys will get together with its respective pairs, like a husband and wife or something like that. But don't confuse with mother and father. But I'm telling you, the homologous pair is 1, 1. 1, 1 is similar type. 2, 2, it's a similar type. 3, 3, it's a similar type. So they're getting organized. So this is called as pairing of homologous chromosomes. Once they start pairing, this process is called as synapsis. What is synapsis? Synapsis is a process of finding your partner and then you lay. See, this is your X, this is another X, and then you overlap. This is one X, this is another X, and then you overlap like this. You just come on each other. Okay, come over each other. So this is once they come over each other, they form a physical connection. Okay, they attach to each other physically. So that is called a synaptomial complex. A complex is means what? A structure that cannot be defined what it is. But what is happening here? The two structures are involved. What is that? Those structures are called as homologous pair of chromosomes. When homologous pair of chromosomes have found its partners, they have come across each other like this. They overlap with each other. They start forming a physical connection. Okay, so this process is called as synapses. And once they form this complex, once they are overlapping with each other, that is called as synaptomial complex. Did you guys understand this? Yes, no, faster? Yes, please respond. Very, very important it is. You guys have to respond to me, especially in this topic, please. Anybody? All right. Okay. Chalo. Now we have the synaptomial complex. The overlapping has occurred. Okay. Let's check. Okay. Yeah. Thank you, Trisha. Okay. You're alive. I thought you guys were missing. Okay. Chalo. So now we have the homologous pair found by the synaptomial complex. Now these pair have been occurred. Now what can you see, guys? Now each pair, I told you that this complex is formed called a synaptomial complex. And since two homologous chromosomes are involved in this, I can call it as a bivalent. What is bi? Bi means two. Valent means, what is valency, guys? Sharing. According to chemists, what do you say? Bivalent means what? Sharing. You can see here, right? This is one homologous pair. This is another homologous pair, and they started attaching. They were gummed together, okay, at a point. And this is called as a synaptomial complex. And after the formation of the synaptomial complex, now when I look at it, two structures have formed something like a valent bond. Okay, like valency, sharing. So there, since I can see that there two are involved, I can call it as a bivalent. So you can say, hold on, paired homologous. Yeah, paired chromosomes are called as a bivalent. Exactly, paired chromosomes with the synaptomial complex is called as a bivalent. Guys, remember, when does a synapsis happen? Zygote. When does a synaptomial complex forms? Zygote. A homologous pairing occurs at what stage? Zygote. Okay. The formation of a bivalent is seen at what stage? Zygote. Four terms. What are those? Synapsis, synaptomial complex, homologous chromosomes, pairing, and then formation of a bivalent. All of them is happening at what stage, guys? Zygote. Please write it down if you don't remember. These terms are much more important than the whole story. Okay, because you can easily recognize the words. Okay, let's go for the zygote. Yes, guys, zygote. Next stage of the zygote is what? Packaging. What is a packaging? It is the longer phase compared to that of your zygote. What is happening, guys? The homologous chromosomes have created some structure, and this structure is called as a bivalent. Once this bivalent is formed, now they go into the next stage. What is that? Here, instead of overlapping one on each other, they come sideways. They just come sideways until a point. So once they come sideways, these are called as tetrads. Why do we call it as a tetrad? Four. Okay, four threads, four thread-like structures. They align beside each other. The point at which you can see that the overlap synapses has broken the synapses. The synapses will end, guys. The synapses has ended. And then with the end of synapses, you can clearly see that these two have come aside with each other. And in the microscope, you can see clearly that they're very, very closely associated, but four thread-like structures can be seen. Okay, so this is called as a tetrad formation, or it's also called as tetrad. But are they separated completely? No, as they move across from each other, as they move from overlap positions to sideways positions, now what they do is we have arms, no? So this is called as an arm. This is also called as an arm. So the arm of one chromosome, let's say that this is the arm of one chromosome, and this is the arm of another chromosome. Okay, as they move each other, these arms overlap with each other. So they are not just overlapping, they're crossing, I can tell. So they're not just overlapping. Okay, they're not just overlapping. And this overlapping is also called as what? Crossing over. So once they have formed this crossing over, they're not just one above each other. Okay, so they're not just one above. So I can tell you guys, is it given here? No. Let's say that this is one chromosome. This is another chromosome. Now, what has happened to these? They are overlapping. So when they're overlapping, let's say that, sorry, Okay. So this is how the overlapping has taken place. What is it? Even I don't know, but you can see that the lines do not go like this. So this is one chromosome, this is another chromosome. The lines cannot be visible. Why? What happened here? You can see here when they overlap, they completely, I can tell it is fusing. So let's say that what is chromosome made up of? DNA. This is also DNA. So when they come in each other, they're mixing up with each other. So they are fusing with each other. So we don't call it as fusing. We call it as something called as recombination. Now this is a very, very important term. What is recombination? See guys, let's say that this is the DNA of one arm. This is the DNA that is present in one arm. And this is the DNA that is present in the other arm. Now, the overlapping has taken place between these two DNA. What is happening here? What is recombination? Recombination is called as bulk transfer of genetic material. What is happening here? Bulk transfer of information is happening. Why is it even required? Okay, how can I recognize recombination? So you're telling that the lot of information is passing between these two, sister arms when they're crossed. Absolutely correct, guys. So how can I find this? How can I see this in physically? Of course, you can just take an example. Guys, can you see you? Just take yourself, look at your face, look at your characters, and now compare it with your mother and father. Some of the characters you have from mother, some of the characteristics you have from your father, but you don't exactly look like your father or you don't exactly look like your mother. Agree with me? Yes. Why? Even though I'm a human being, even though I'm the product of my mother and father, why don't I exactly look like either of one? I'm not only just sharing their characteristics, I also have new characters. Maybe I'm taller, maybe I'm darker, maybe I'm more intelligent or more dumb, whatever it is. You can see entirely new characters are coming. So how come these new characteristics appear in the next generation? Is something happening? Definitely happening. It's happening at the genetic level. We call it as genetics. So here what is happening, guys? The bulk transfer of information takes place when the two sister chromatids, okay, when the two arms cross. I will not mention whether it is sister or non-sister chromatids. Let me tell you, when the two arms cross, what is happening, guys? A recombination nodule is formed. When the recombination nodule is formed, okay, recombination happens. What is recombination? That means bulk sharing of information happens. This is a very, very crucial step. So this crucial step is giving you all the beautiful varieties you see in your classroom, in your human beings, in the nature. So this is happening. This is called as formation of recombination nodule. And I told you what is the meaning of recombination. So let's get back here. The bivalent chromosome, what they did, they split, they form tetrads. After the formation of tetrads, we saw that they are not completely separating, but one arm of chromosome overlapped with another arm of chromosome. Now, if you see this arm, let's say that this is the centrosome. This is one sister chromatid. This is another sister chromatid. This is another chromosome. Okay. What did I say this? This is called as what? Sister. When do I call something a sister? When my parents give birth to two children, I'm a brother, and if it's a female, that is a sister. If it's my cousins, they are cousin sisters. They are not called sisters. That's why this is called as a sister. This is also called as sister. But when two of the sisters from two different chromosomes are involved, what do we call this? This is called as non-sister chromatids. Okay. What arms of the chromosomes overlap during the packaging stage is our question. So we can see here there is an overlap happening. So this is a small word we forget. What is that? During the packaging stage, after the bivalent, the chromosomes organize into a tetrad fashion. So once the tetrad formation is done, the appearance of a recombinant nodule occurs at the crossing over between who? Non-sister chromatids. Non-sister chromatids. Did you guys understand this point, please? Can you confirm? Yes, no? Did you understand? Okay. Even though it is 1, 1, but one has one sister chromatid, other has another sister chromatid. So they crossed over each other. So since the crossing over has happened between two different sister chromatids of a homologous pair, now I call it as the crossing over between non-sister chromatids. So you should understand this point. Okay, fine. So you guys have told yes, I believe you, and I'm moving ahead. So recombination is complete by the end of what? Passivity. So we have seen that what is recombination, and it is beautiful concept. You'll study more as you come to a +2. Okay. Now it leads to what? Diplotin stage. So now we know that the chromosomes are in this particular position. At this stage, where the recombinant nodule has formed, recombination has occurred. Now what happens here? Once the recombination is complete, now the dissolution, or that means the breaking of the synaptomial complex has to happen. Where they have attached, where the recombinant nodule is created, that should be broken between them. They have to be separated physically. So the dissolution happens. So the bond or the valent condition between them, it gets dissolved. The chromosomes, what chromosomes? The homologous chromosomes, which were attached to each other at the recombinant point, they become free. Okay, the bivalent what? Separates. Like this XX overlapped condition 2, they become what? Like this. They are separated now. Once they are separated, they come back to these X-like structures. Okay, so this X-like structures, they are separated completely, except that still the crossing over position is there, no? Recombinant nodule dissolves completely. You understand this, guys? Recombinant nodule means what? The complete fusion. That complete fusion has been removed. You can still see that this arm is separated, like from complete. Sorry, if I'm repeating this, if from the complete fusion position, they have been separated like this. But are they pulled apart each other? No, sorry, they're not pulled apart each other. So they have been bivalents have been separated. Okay, except for what? Except at the crossing over. Except at this junction, they still overlap. Okay, but they're not separated. So when they're still overlapping but not separated, this condition is called as what? Chiasmata. What is chiasmata? When does chiasmata occur? Chiasmata occurs by the end of recombination. Okay, when in what stage does the chiasmata comes? Chiasmata comes at the diplotin stage. So when the chiasmata has appeared, that means that recombination is complete. The bivalents are separated. Okay. Now, from the completely fused condition, the chromosome has gotten back its shape. It has come back to its original shape. Now, they are not completely separated, but they still overlap at this point. These X-like structures, X-like crossing structures are called as what? Chiasmata. Okay. Now you can see that in many of the organisms, including human beings, when the human beings are babies, like for example, females, they produce what? When they reach age of 13 or 14 during the puberty, they undergo ovulation. Even the boys undergo spermatogenesis. But especially you can see an example. For the formation of ova, you have to undergo oogenesis. Let's say gametogenesis. So during this phase, when they're baby, all these cells which are responsible for production of ova, they undergo meiosis 1, prophase 1. They come to the diplotin stage. Okay, diplotin stage, and they stop. When they're fetus, when they're still in the mother's womb, the moment they know that they're females, obviously it's genes or something has happened. Okay, so once the female fetus is developing, once their ovary starts developing, you can see that the ovaries responsibility is to produce what? Ova. Okay, so now the cells which are responsible for producing ova, you can see that they also have to undergo meiosis because they have to produce gametes, gametogenesis. So gametogenesis means meiosis. So they undergo meiosis 1, they undergo prophase 1, they come up to the diplotin stage, they do not continue further. Reason being, don't know. It's like evolution. So this can last, for in human beings, it can last up to 12 to 14 years. So in any cell cycle, which is the most dormant or hibernating or inactive stage in all of cell division means, what is your answer, guys? Diplotin. The cells arrest at one cell stage for from some weeks to certain years. Which cell stage is that? So you should tell that it is meiosis 1, prophase 1, diplotin. Got it, no, guys? Always be specific. Meiosis 1, prophase 1, diplotin stage. You can see that the cell cycle can arrest from few weeks, from few months to some years. So just example, human girls, females, you can see that the arrest is for 12 to 14 years, they can remain quiet. They will not go further. What is our further stage from diplotin to diakinesis? Is it clear? So the last stage of prophase one, okay, is diakinesis. What is diakinesis? I know that there is X-shaped overlapping. The overlapping is just like, the arms are one above each other. So this overlapping is removed. Terminalization of chiasmata take place. Once the chiasmata is removed, okay, now what will happen? The chromosome gets completely condensed. Now the mitotic spindle fiber starts originating. Correct? No, this is what stage? Prophase one. Correct? No. What happened in prophase? End of prophase. I told you. You can see that from the centromeres. You can see that the spindle astra is developing. It is developing faster. Okay. And it is reaching towards what? Your chromosomes. Now it comes almost, and now it has to attach to these homologous chromosomes. Okay. For its separation. Now, by the end of your prophase, you can see that the nucleolus is become disappeared. Nuclear envelope also disappeared. Cell organelles also disappeared. Did you guys understand? I'm sorry if I prolonged. I hope that this eased your understanding with meiosis one, prophase one, which is a very important phase. Please go through the diagrams. Words. What are the words? I'm sorry, I just think a minute or two. Okay. Liptotin, formation of chromatin fibers. Okay. And the chromosome formation takes place. Nucleolus enlarge. Zygote, what happens? The chromosomes are there. They start pairing with each other. How pairing? Homologous pairs get to each other. Once the homologous pairing starts, this process is called as a synapsis. So once they are paired, they are attached with each other. They overlap with each other. This is called a synaptomial complex. Once the synaptomial complex is formed, what guys? So you can see that there are, hold on, somebody has asked a question or something. Uh, can you show slide for diakinesis? All right, hold on. I'm just revising. I'll show you. Okay. So what happened, guys? Once we have this synaptomial complex, we know that this is having two chromosomes involvement, so we can call it as a bivalent structure. So bivalent synapses, synaptomial complex, and homologous pairing all comes under zygote. Next comes up packaging. So once they are paired, you can see that the synapses is converted into a tetrad condition. They're from overlapping to sideways position. So they come to tetrad condition. Now at the tetrad, the two non-sister chromatids, they start overlapping with each other. So they start creating what? A recombinant nodule. So once they form the recombinant nodule, the overlapping happens, or it is also called as a crossing over. So once the crossing over is done, the reason for why this crossing over is to complete the process called as recombination. Recombination, crossing over, recombinant nodule, and tetrad, all of them come under Pachytene stage. Got it? Next comes a diplotin. We know that the recombination is complete. Once the recombination is complete, recombinant nodule has to dissolve. So the recombinant nodule is dissolved, but the bivalents are separated. They start moving away from each other, except at one point where the recombination has occurred. Which is completely fused condition, they have come across the normal condition, but they are not completely separated. So since that stage will mark that stage where you can see the X-shaped structure at the crossover. So that point is called as what? Chiasmata. Chiasmata will show the end of recombination. Got it? No. So we know that diplotin is the longest waiting phase, we can tell. So it can last for months to years. Example, oocytes in human beings. Next one is diakinesis. I think diplotin guys, don't worry about the diagram. I will get the diagram, very detailed diagram. So which comes in your NEET examination. I'll show the slide next class. So you can just make some notes on it. Okay. Next one is diakinesis. So we know that the chiasmata is completely terminalized. The chromosome is highly condensed. Now the aster formation is happening. The spindle fibers are reaching for these chromosomes. You can see the nucleolus and the nuclear envelope disappear. Very, very important phase. That's why I have taken enough time to explain you these guys. I hope this class helped you. Okay, we can end the class because we're almost time. Before you leave, please give a feedback. Okay, just tell if you have doubts. If you're satisfied, we can correct this in the next class, guys. Thank you very much for attending this session. If you have any doubts, I'm open. You can step back and ask your answers. I mean, like questions for a few minutes. I'll be here, guys. Thank you. The day all of you answer, like you respond to this poll, I'll be the happiest guy, I feel. Yeah, I'll be really happy. Yeah, so that will be my most satisfying class when I get all my students talking and being here for me. All right. So please feel free, guys. This is the only way we can get to know each other. Already, a world is such a place where we can't see people and meet people. Okay, thank you very much for your review, and I hope we continue the quality of this class even in future. See you guys. Have a nice day. Bye bye.